Okay, we're going to continue chapter 11. This one's got a little bit to it. If TV science was more like real science, first box, serial killers would have plenty of time to get away. Is it here's TV, quick, run a PCR DNA analysis on this sample. Yeah, that's an overnighter. Is it, second one, special agents would never figure out who the villain is. We reconstructed this image from a 4-pixel photo. Turns out that's theoretically impossible. Myth debunking would never get past peer review. That's the third one. What do you mean one data point is not enough? What's the control? And robots would never take over the world. It was working a second ago. So we just talked about development. Here's spiral cleavage, early, just a few cells. Little cells, big cells, the little cells move into the middle. Eventually they wind up kind of pushed to the outside of the developing blastula. Here, remember I said it looks like a radius, well, a diameter, radial cleavage. Once the hollow ball indents, the mouth in a protostome is going to form from this. The anus in the deuterostome is going to form from this. The mesoderm forms at the junction of the two. The mesoderm forms from outpockets. There'd be more than these two in it. And at this point, or very close to this point, actually even earlier, you could pluck a cell out of an embryo and its fate, its genetic future, what it's gonna do, what part of the body it's gonna be, is kind of laid out already. It's already started toward the road of differentiation. Whereas here, the road toward differentiation is laid out much later, and it's much more influenced less by epigenetics, where how and when particular genes turn on are already kind of laid in, and more on environmental responses, where the cells in a neighborhood influence each other to do different things, which is one of the things about our stem cells that that allows us to take cells that have the potential to do anything and put them in places to replace damaged tissue. They'll read the environment chemically and become those kinds of cells. I would assume there's some things that you might be able to do that. If we were deuterostomes, maybe that they have stem cell populations, but I don't actually know that. I've never seen anybody talk about potential stem cells in protostomes. If we were, arthropods, then we probably would know all that stuff. We talked about this way back when. That living things are internally organized, and we talked a little bit about the levels, so this is going to be a revisit of that. Inside the cell, there's an hierarchy and an organization. That's protoplasmic level. Cellular includes not just the cell, but cells around the cells. That's what I was talking about before with development in deuterostomes. And it also includes influences of things like tissue fluid and non-cellular components around the cells, which when we get to the tissues in a couple of minutes will make a lot more sense, because there are some tissues that are very much defined by non-cellular elements. There's a cell tissue level and a tissue organ level, I'm going to write this down and then go through it. Tissues are collections of cells that have certain broad features in common. We're going to go through the animal tissue types. Second semester, we'll go through them in a lot more molecular detail, but here we're going to kind of go through them in kind of just broad details. Those tissues wind up getting integrated into organs. Organs and structures, what's the difference? Well, welcome to biology. An organ is kind of a localized collection of tissues that do one or more particular jobs, usually more than one particular job. Very often if it does just one job, it gets reduced to the term structure. And sometimes when it does a couple of jobs, it's generally called a structure. But you will see that vary from book to book. Is that most books would consider a bone a structure, but there's a lot of books that consider bones an organ. Your trachea is generally, your windpipe is generally considered a structure. I've seen books that call it organs, where they call almost everything an organ. So there's no clear defining level that everybody agrees to. And then you put a bunch of these together doing some sort of real basic job, like respiration or circulation or reproduction. Or slow control, fast control, things we're going to talk about in the following chapters. And now you're talking about organ systems. Now, a lot of this, when you go through the books about it, is very vertebrate, backboned animal centric. Whereas we're going to try to talk more in general terms about animals in general. But my overheads are much more vertebrate centric. We actually do a whole lab on this in the second semester. Here's an intestine. This is just a little bit of the wall, the lining of the intestine. This is the outside, this is the inside, this is where all the digestion would be happening and the absorption would be happening. Remember I said that there's projections into the space that increase the surface area? These cells would have microvilli on them, which would increase their surface area as well. But you have layers of tissue, and each tissue is there for a particular part of what that tissue does. You've got an internal lining, an epithelium. We'll go through the tissue types in a minute. In this case, the epithelium is doing two of the jobs epithelium does, actually three of the jobs epithelium does. They secrete materials into the cavity, things like mucus to protect the lining, and digestive enzymes, some of which come from the pancreas, but some of which come from the lining. And they also form a barrier against the digestion actually digesting the lining. That's what producing the mucus does. And those are three things that epithelium does. Now this epithelium doesn't really do any of those. Is that this is the outer epithelium of your intestines, and it basically is an edge. And that's, its job is to say, okay, this is where the intestine stops and the space around us in the abdomen begins. And that's an important function, but that's all this epithelium does. And you got layers of muscle, the squeeze that pushes food through. Any kind of structure tends to be held together by a type of tissue called connective tissue. So what makes these projections hold their projection structure is connective tissue inside. And connective tissue does a number of different things that very often are sustaining structure one way or another. So we will go through these, but these are 4 of the possible 5 types of tissue that we're going to go through when we go through this list. When we revisit this in the second semester, it turns out plants have tissues as well, some of which have similar basic functions to our tissues, and some of which don't, and some of which perform very differently than our basic approaches. The study of tissues is histology. So if you have a biopsy, it's a histological specimen. It's going to be looked at on a cellular level. If it's a biopsy, most of the time they're looking for cancerous changes. But there could be other things that happen too, that you can see on a cellular level. And people study that. So now we're going to go through the 5 types of tissues. Not all the books agree that there's 5. Oh, crap, we're going to stop.